Very much for the introduction. Yeah, let me reiterate, I have much more fun if you ask questions during the talk than just after, so please go ahead. I guess you need a mic, but I really prefer this. The other thing I want to start with is a shameless plug. We are looking for a tenure-track assistant professor candidate, in soft condensed matter. The ad is not quite out. It's still in the bureaucratic mills of UC Berkeley, but hopefully within the next couple of days it will be out, and it's open to both experimentalists and theorists, and it's also broadly construed soft condensed matter physics we're interested in. So this can be biological, but doesn't have to, not at all. It can be non-biological, nonlinear dynamics, non-equilibrium statistical physics, and active matter, of course, as well. So I want to put it here for two reasons. First of all, that candidates know this, but also if you're a PI, more established, and you know excellent candidates, please give me some hints. I'm really interested in knowing, especially about the field of active matter, and who do you think works at the very forefront of this field, so that we can reach out to them as well. All right, so now about my talk. So this will be a talk about active matter where activity is not introduced by motion, right, where some chemical free energy locally available is converted into motion, but the activity exclusively comes from proliferation. And in fact, the motion will be completely passive and not break any time reversal symmetry. It doesn't look like I'm actually dealing with this based on this picture. I want to show you the picture first of all because these are beautiful islands. But actually just 4 of them are real. The other ones I created by very simple copy and pasting and mirroring, like so that you don't see where I connected them. But this way it looks like a lattice. And I want to show you that because if we had lattices like these, we could do beautiful macroscopic experiments. But one point of this will be, it's very hard to do experiments on the macro scale, even though very, very interesting questions have already been asked before I was born about systems on the very large scale. And that's the topic of island biogeography. So essentially the talk will be inspired by island biography, working on these scales and bringing the questions down to the micro scale where I think they are both answerable, both experimentally and theoretically, and they are relevant, I think. All right, so let me first say a couple of things about island biography. Right, so this was a field created essentially in 1967, before I was born, by these 2 people, E.O. Wilson and MacArthur, that very humbly wrote the first book about this field and already called it the theory of island biogeography. It is very inspiring. So they is asked, in the simplest form, a question about biodiversity. How many species fit on an island? That's the simplest version of the question they asked. In principle, this is a very, very simple question about a very complex system, and the answer could be complex, but they said, well, maybe it's a simple competition between few forces, maybe just 2 forces. So in the simplest case, they would say, well, new species are brought to such an island by immigration. So some kind of animal, floats on some driftwood and is brought to the shores of this island. So this is one way a new species might arrive and then establish there. That's immigration. The other one would be like if you have a finite population of a certain species on this island, at some rate this will go extinct. Of course, the larger the population, the smaller this rate is going to be. And then they said, how could we think about these forces in a simple toy model? And they drew pictures like these, where you would have here 2 curves. One is the blue curve, which shows you the immigration rate, how many new species come in, as a function of how many you already have on the island. And clearly, the more you already have, the fewer will be out there that you can discover. That's why generically this should go down. On the other hand, the red curve tells you something about extinction, which should go up with how many species you have on this island, simply because you have more possibilities of a species to go extinct. And somewhere these two curves cross, and they would say maybe this is kind of where the equilibrium would be. This would still in itself be not very valuable because, where did you get these curves from, right? This is very complex to compute. But kind of what was inspiring to the people in the field was, well, everything else equal, if we just change the size of the island, we can intuit what should happen to these curves. The immigration rate probably shouldn't change, but the extinction rate should go down, and therefore large islands should have more species. And this is one type of argument they made. And this was pretty inspiring to conservation biology because people realized we can apply this not only to islands potentially, but to any kind of ecosystem that is surrounded by something else so that there's a boundary between given ecosystem and that's something else. And we can make statements about this ecosystem then. And in particular, people were interested into natural preserves and how they could be designed, or national parks. And so the battle cry that then emerged by Jared Diamond was that a single large reserve is better than several small reserves, just based on these relatively simple theories. Well, and then it became really serious, right? Should we adjust our policy based on such theories? And it created a huge debate, which even has an abbreviation, the SLOSS debate. And there's a beautiful book about this ecological fight and gang fight that emerged. Because people argued, well, you make lots of assumptions, you focus on equilibrium, why should it be equilibrium? You also assume, you compare islands where you think everything else is equal, but islands are so variable, you can't just make these statements, right? And the problem is empirical tests were really, really hard. They were made, but going in both directions, and somehow this whole field then dwindled over time. I think we actually now are in a great stage to revisit this, even on the macro scale, because there is now an enormous amount of data on remote sensing, and perhaps some of these predictions can be tested more rigorously just with the amount of data that we have. But my talk will not be about this, but on kind of the micro scale, and I'll get to that in a second. I also wanted to say that out of this, what really emerged and became very important to ecologists and theoretical ecologists was the insight that kind of emergent phenomena like diversity and evolution really are scale dependent, and we have to look at our system as a function of scale, and new phenomena emerge as you go to larger scales. There's a beautiful essay in ecology about this, which is essentially the ecologist version of More is Different, which I'm sure many of you have seen. And ours was earlier, I should say. All right, so what I want to talk about is essentially to ask similar questions about an island biography for microbes, right? And first of all, if you think about us and you ask, okay, where do I have to go to find an island for us? Well, it has to be a relatively large island, which you find out there on the ocean, but it's not so abundant. If you think about microbes, on the microscale, a simple dust particle could form or act like an island, or the reverse, a little cavity, right? So I think effective islands are really ubiquitous for microbes. And I also think it really matters. That's going to be the next point, because microbes have been shown to colonize preferentially on particles as well as cavities in the soil and also in the gut. Right? So here are examples that might matter to our health. On your skin, my skin, we have these lots of these cavities here, sebaceous glands, and they're filled with microbes, with bacteria, very, very stably. And in our guts, we have colonic crypts, which are also stably colonized by bacteria. And stable in the sense that once you, in experiments, bring in bacteria of one type, and then you try to bring in another type of bacterium, let's say even of a similar type, and you label that new bacterium, it's actually very hard to colonize these pre-colonized crypts, right? So in that sense, this colonization is very stable, and that's also called colonization resistance. And so in what I'm going to show you, I show you some experiments to explore how this colonization resistance arises, how we can quantify it, then to make essentially statements back about these island biogeographic questions. Right, and yeah, go ahead. Yeah, please, thank you.
So is it important in this resistance to colonization, is it important that these invaders have to come in through a small entrance or something? I mean, let us say you don't have to guard the whole region, you only have to guard the entrance to prevent entry, right?
That's right. I mean, I'll get to this question when I precisely describe the difference between coming from the outside and from the inside, which a mutation would do. Right? You might also ask about what about the invasion of a mutation, a mutant that comes from within, like you infiltrate the population. And there's actually not such a big difference, as it turns out. Thank you for the question. Keep going. Okay, so my grad student at the time, Yuya, he's now a postdoc in Germany. No, actually, he turned into an assistant professor just recently, going back to Japan. Yuya, he came up with a microfluidic device to study this scenario. Right, this is a very simple device where you bring in these bacteria and also mostly medium through the supply channel, and then as the bacteria float along, they're exposed to these cavities and can potentially colonize them, and that's what we can explore. And you might have seen many such devices, for example, the mother machine, I'm sure you've seen it. Those exist of cavities that are very, very narrow, so that with bacteria it looks like a sausage factory. Here they are wide, so they can overtake one another. That's one difference. But otherwise, also such devices with wide channels exist. The main innovation is that Yuya systematically varied the length scale of these invaginations to see, what's the difference, what depends on this length scale. And because it looks like a pan fluid, we call it the microfluidic pan fluid. And the stuff that we want to study in there, in principle, it would be great, seeing the talks in the morning, to have something like hexbugs that can proliferate. I haven't seen them yet. I think that's one of the big frontiers in engineering, to introduce proliferation to these gimmicks. That would be amazing. I would like to stuff it into my device. But in the absence of having those, we need to refer back to good old nature. Look at this name already. And you zoom out. Acidobacter indonesiensis. I show you mostly movies involving this bacterium. We have done several others where we see similar phenomena as well. This one here we took essentially from our friend Will Luddington, who studies the phenomena inside the fly gut. So this is native to the fly gut, this particular bacterium. Importantly, this bacterium has no activity in the motility, right? So it's purely passively moving and diffusing effectively. All right, first let me show you some steady state images here. As you come from the small end of the cavity, where the cavities are relatively small, you see they are largely empty, and then at some point they start to be filled. And the region over which they are filled is kind of has a characteristic length scale, which becomes important in a second. So what's going on here first intuitively is the following. Right, the medium brings in nutrients so they can grow. There's lots of division going on here, essentially no death, no real death. But there's an effective death occurring, which is when these bacteria randomly diffuse up and get caught by the flow lines, then they get carried away. And both of these forces scale differently with the size of the population. That generates the length scale effectively, which is the critical length scale over which we first see colonization. You can mathematize this relatively easily. The simplest version is to ignore all the coupling to the hydrodynamics, which is really interesting, but to say, okay, we study the population dynamics just effectively one-dimensionally here along this vertical line, which I call x, and then I say the rate of change of the population density c as a function of position and time t depends on just two factors. One of them is diffusion, and the other one is growth with a constant growth rate r. Right? And then this linear PDE needs to be supplied by boundary conditions. First of all, no cells should leave through this closed end of the cavity. And the other one is an effective absorbing boundary condition. Somewhere we know the density goes to 0. That's what we call x equal to 0, even though effectively this is created by the flow lines, which we don't model here. But given this, you can do a normal mode analysis, of course, and find that the empty cavity is stable if kind of this population length is smaller than a critical length. It has to scale by dimensional analysis in this way, square root of diffusivity and growth rate. And you get also then this prefactor here. This is something we can test now experimentally. You can put in, you can independently measure the diffusivity of these bacteria, which is roughly the diffusivity of a ball of radius 1 micron, which is what you expect. The growth rate you can also measure, and so you get a length scale of about 50 microns, which roughly agrees with the experiments. But more importantly, you can also do time-lapse movies and then average these time-lapse movies to get like a cell density as a function of position. And close to the critical point, the density as a function of position also agrees with what you expect, this cosine function, except for the low density part, which is this region here, where really the flow and the hydrodynamics matter, which we don't describe here. All right. This, however, is a theory that's completely linear. It only describes the stability of the empty cavity. And so at some point it becomes unstable, but what happens then? Well, this would predict exponential growth, and that has to stop at some point. But how does it stop? That depends on the figuring out the right nonlinearity, technically. And so let me first show you just what we see in the experiments. So now we go towards larger cavities, and you see that the density of these proliferating bacteria is increasing. Right? It looks like lots of ants in here. And at some point, this dark phase appears. And if you look closely with a confocal microscope, you see that the bacteria in there are touching. So they are effectively jammed. And that's kind of surprising that there's this jamming transition. It's in fact discontinuous. You slightly change the size of the cavity, and this phase appears. I'm going to convince you that what's happening here is essentially emergent traffic jams and positive feedback. I'll get to that in a bit. I first want to show you dynamically how they form. Here you see these kind of shock fronts running through. And as you get closer to the critical point, it takes longer and longer for the dense phase to form, which is really reminiscent of kind of a critical slowing down.
Your time stamp is hours?
Yeah, here you see the hours. It's now 35, so many cell divisions it takes for near the critical point to establish. All right, so now it turns out we can measure the immediate instinct, perhaps, of most would be to introduce a nonlinearity in the growth rate, right? Because when things get denser, they compete for food, and so maybe the growth rate goes down. But we can measure that independently. And it turns out the growth rate doesn't practically change, at least on the scales that I've shown you. So then what has to happen is the nonlinearity needs to enter through the transport term. And so what we need to think about here is the collective diffusion coefficient, which is this coefficient here as a function of density. How does that depend on density? Well, that's in general hard to say. From colloidal physics, one knows that this can be written as a product of two terms. One is a purely static term, which is essentially the osmotic bulk modulus as a function of density. And the second term here, gamma of C, is a mobility, which is essentially related to the sedimentation velocity of these colloids as a function of density. And Bulk modulus generically goes up with density, but mobility goes down. So what does the product do? It's really not clear from the outset. But what we can do is that this equation, if you look just at the steady state, there's an exact mechanical analogy we can exploit, where we can formulate effectively like a density-dependent pressure, which is the integral of this collective diffusivity over density, and then write the steady state equation in this form where the gradient of the pressure represents effectively the particle current, right? And then you map the equation up here to Newton's equation of a particle in an effective potential U, and this mapping allows us for any function d of c to ask how does the steady state profile look like, and essentially then explore for what kind of functions d of c do we observe this kind of phase transition. And it turns out what you need to have is that the collective diffusivity first goes up and then sufficiently strongly down. I can't say it more precisely other to go through a lot of math. But the negative slope is really important because then, well, you get essentially a positive feedback where you have a density increase, less diffusion, less outflow, and that can only stop once the thing jams. Right, so what could cause this negative the negative slope of the collective diffusivity? Could be many things, including cell stickiness. So we asked, do we observe this even for the simplest case of just proliferating hard spheres? And there you can go to the literature, and we have for both terms the mobility and the bulk modulus, osmotic bulk modulus, quite good approximative formulas, and you get a hump-like shape. So this includes also all the hydrodynamic interactions that are present. And this hump-like shape is enough to indeed drive the transition that I mentioned. It's just effectively a fold bifurcation, which you see here, the maximum packing fraction at the floor is a function of the size of the population, has this shape. And so as you increase the size of your population, at some point you jump to the jammed state. And in fact, when you then go back, you jump at a different place. So there's also hysteresis, which in the experiment, in fact, we can also show to be the case. All right, so there's a question, yes?
So why does it only depend on the height of your colony and not the depth of your trap?
Could you say that again?
Why the length that you have is just like the width of the colony, right? L?
It's the length of the colony, yes.
Yeah, so why don't you have the full depth? Like why? Because
why is it not the full length of the cavity?
Yeah, I would expect that like what is above matters because you have like less.
Thank you for the question. That's important. So this goes back to where is our absorbing boundary here? This depends on how large is the flow through the supply channel. The supply channel, the larger the flow, the deeper the flow lines go into the cavity and sweep out everything that's in there. So essentially there's an emergent absorbing boundary that depends really on the hydrodynamics, which is a more complicated problem, which is not part of this story. It's an interesting one to explore. For us, it's actually good because we can tune the flow rate to tune the effective size of these cavities. All right? Thank you for the question. All right, so now to this question of colonization stability. So here you see a device that's pre-colonized with kind of, let's call it the wild type bacteria. They are gas-like, and here essentially jammed. And when we now bring in an invader, if it's of exactly the same type, just labeled, we see never any invasion. It seems to be really, really hard. So we take a strain that we give an advantage. In fact, we use a strain that is resistant to an antibiotic, to tetracycline, which prevents growth, right? And this also is interesting, to ask the question in our guts, how easy is it for an antibiotic-resistant strain to colonize these cavities? All right, so in the first 21 hours, you see, when we bring in the tetracycline, what happened here, let me go back. Essentially, these jammed-like, this glassy state here kind of melts and turns into a gas, but still after 21 hours, we see perhaps here the first green cells. On the second day, some invasions occur, and they start to be successful. After 5 days, we do see a highly variable kind of state of invasion. This panfield was heavily invaded, the other ones differently. There's, I think, a very interesting collective dynamics here. Once you invade one cavity, it acts like a source for the surrounding ones. But here I just want to point out that in no case do we see an invasion of this very jammed and crowded cavities. And the reason is that there, the now self-diffusivity of the bacteria is very, very low. Not the collective diffusivity, but the cell diffusivity, which makes it very hard for an invader to diffuse or to infiltrate kind of this population all the way up to the cavity. This is something, that the self-diffusivity is low, can be measured. Here in the gas-light case, where the self-diffusivity and collective diffusivity are essentially the same. But here in the jammed case, the self-diffusivity, meaning the diffusivity of a labeled particle, is much, much lower, essentially just 1 micron per cell division, micron squared per cell division.
Oskar?
Yes.
These are presumably rod-like rather than spherical bacteria.
Yes.
And so, or do they acquire pneumatic order by virtue of the walls when you jam them?
Yeah, we haven't seen this yet, and I think that's an interesting question. I think proliferation essentially makes this constant injection of new degrees of freedom makes it really hard for them to order pneumatically. At least we don't see it, right? I don't have a theory for that.
Have you looked at cross-polarizers? With cross-polarizers?
Oh, we haven't looked at that, no. Good suggestion. Thank you, David. Yeah. Is that okay? Can we add these minutes?
Some kind of a plume or something in the middle?
Yeah.
In the right picture, is that structure down through the middle?
Yeah, good question. So here we looked for a case where we have both types present. So the dark stripe is the wild type and the green one is the resistant one. And this is in the absence of the antibiotics, so they are neutral. And here we just look at these boundaries and how they fluctuate in order to get an idea of what the self-diffusion is. Because for us it was very hard to measure that in this very dense gene. So essentially it's a tracer experiment to get at these fluctuations. All right, then I wanted to also tell you a bit about selection, right? And this question, what if you don't come from the outside but you're already within? And how hard is it then to actually take over? So here's a scenario where we colonize at the same time with both types. We get lots of cavities where both types are present, and then we turn on selection by turning on tetracycline, bring in tetracycline. What happens is a dynamics like this. Ultimately, what do you expect? Green takes over, but it happens in 2 phases. The first one is actually both types go down, right? You select against the wild type, but the green also disappears. How is that happening? How can that be? The reason is that they rely on each other to be filled up all the way to here, because if you take away the dark strain, essentially also green starts to increase its self-diffusion, or its diffusion, and then will disappear as well. That's what's happening here, and nothing changes about the fraction of green versus dark. That only changes, so as long as we have this glassy dynamics, we have essentially no selection, green doesn't take over. But then at some point it turns into a gas, and we have self-diffusion and mixing, and then they compete, and exponential takeover occurs. And so let me conclude. So looking at this battle cry of island biogeography, a single large preserve is better than several small. Certainly not true for our case, because the largest cavities actually have the least diversity, right? Because they are jammed and nothing can go in at all. So it really depends jammed or not jammed, in our case at least, population stability results from a collectively generated infiltration barrier here. I think there's potential for evolutionary adaptation, both of the host, who would like to enrich perhaps certain bacteria, and also for the bacteria to adapt to the situation in the gut. I had the story about selection. And finally, there's really, I think important take-home is that there's extreme scale sensitivity near phase transitions in the system. So it's very important to make these experiments really scale dependent. I'm essentially done, right? Okay, let me just show you kind of as an advertisement, as a plug for this paper where we studied now really spontaneous mutations occurring in this very dense jammed state, right? We engineered here bacteria that can switch from red to green. I hope you're not colorblind. But there you see these patches, and they come in wide ranges, size ranges, and they have a particular broad spectrum of sizes. This was actually just neutral mutations. We're extending this to non-neutral ones. And I would like to extend this in various directions, systematically varying not only the length, but also cavity shape, pan-fluid structure, etc. Also look at communities to be formed, also bring in phages. There's so much fun here. And lastly, I'd love to do this also in a more controllable way, not with bacteria, but maybe with molecules that can proliferate. So, for example, with DNA, if you are able to do a PCR cycle in a microfluidic device, maybe we can look at the same questions there. With that, let me thank Yuya for really driving this work. He's standing on the shoulder of several others in the lab that have gone on to other places. And I'm looking for a postdoc, also, theoretical as well as experimental, if you're interested. And otherwise, thank you for listening.
All right, thank you, Oskar. Vincenzo.
Hi, Oskar. I was curious about the last slides where you showed
this
that,
one.
Maybe a couple, that beautiful interface, here.
Yeah.
Have you looked at the statistics of that interface and how, I can see a lot of breakup, but if we forget the breakup, is it somehow altered by the interesting mutation? Is there a feedback of those mutations on the mechanics?
I think in this case, no, because these mutations are completely neutral. So this was our first step to engineer kind of a switch, which just changes the color, but is otherwise a passive tracer. So I don't think there's a feedback with them. This is also a different, I should have said, this is yeast, so it's a different microbe. But nevertheless, these fluctuations are also strong, if you remember, perhaps in these pan-fluid devices, and we haven't characterized them in detail yet. So there's really, in the pan-fluids where we see the jamming, there's also actually a transition from within the cavities from the jammed, very dense state to then a dilute state, and the interface there is also extremely noisy, and we don't understand that yet.
Thanks.
Yeah, very interesting talk. So when you showed that this green mutant invades, like across different cavities in your pan fluid, can we think of this as a kind of discretized version of an expanding Fisher wave in one dimension, or are there potentially some alley effects or something like that?
This here?
Yeah.
Yeah, this is also something we're exploring. We don't know. These were very long experiments, so we haven't done. So we have very rarely done images here, so we don't have kind of the time-lapse movie of how this really was created. But if you just look at the statistics of how many cavities were colonized here versus there, it's clear that there must be correlations. The hypothesis is, as I said, you first establish a foothold somewhere, take over a cavity, and that is kind of then a source for more invaders. And then, it could potentially be like a wave spreading from there, but it's not something we have yet explored.
Right. Yeah, I guess I was just interested if you're definitely going to add some inoculum from the previous one to the next one, but whether there's also additionally something like an alley threshold, where you need say a critical size of the inoculum for further expansion or not, and whether that can potentially halt the expansion as you...
I think for an empty cavity, there's something like an allele effect. So I should say, perhaps explain what that is. That just means that your effective growth rate increases when your density increases. And something like that exists here because of this collective diffusivity, right? If it's empty, you're diffusing rapidly and your rate of being lost is very high. You need others to slow you down. This is something we saw in this selection movie. So that is there. Yeah, so in principle, that could matter for the late stages, I think, of the colonization, where it really then kicks in this positive feedback.
Thanks.
So did you conduct PIV to see if on the mouth of the notches you get vortices or not?
You have a question about vortices where?
On the mouth of the notch, like since the fluid is flowing, it would lead to like a small vortice.
I don't think in this system here the dimensions are such that we don't yet observe vortices, right? So, but they're interesting hydrodynamic instabilities that we're exploring. So if in the z direction you increase the height of the device, then you get an instability that's called lid instability, where you then indeed get vortices within these cavities. And one question is, do they form like breathing chambers? Can you have like one type per vortex take over? Establish per vortex. But in this system, the hydrodynamics is much simpler. There's no vortex.
And also, at what point in time do they start forming biofilms in these systems?
These bacteria don't.
Don't, okay.
It depends on the bacteria.
Thanks.
Well, so it's a lovely talk. I just wondered about this, a nice pan flute analogy. As you probably know, the tubes on the pan flute are logarithmic, as are the notes, the frequencies on a piano. This is linear, and I wondered if you made your crypts logarithmic, literally like a pan flute, would you get a different result, depending on to the extent that these crypts can communicate, and exponential growth is logarithmic in the right variable. So, have you thought about that?
No, I haven't thought about that. Like, the analogy goes a bit further. Like the normal modes that we have are also like similar to some extent, like what you expect maybe for a pressure profile within a real pan flute. But actually, I didn't know that there's a logarithmic dependence of the
organ pipes, so they all have the same diameter, they'll be logarithmic.
All right, but I think really there is essentially this is mostly a single cavity problem for most of what I've shown, except for these ones. That's where for the first time the different cavities talk to one another. Yeah.
I want to be a little troublesome to go back to the sloss debate that you brought up at the very beginning, because it seems indeed, at least for organismal scale, there was or potentially the prediction that there would be more diversity in larger islands. And showing here, as well as also with the observation that colonization resistance kind of works against that. So how does all of this tie back into the original debate on, especially also since organisms and islands don't have this concentration-dependent diffusion mechanism. So can you...
Yeah, so I don't want to make a claim about, big islands. So the idea of the talk is really take it as an inspiration to think about microbial island biogeography. I don't think our results here tell us anything about what's happening in kind of the big world, other than there can be surprises that you didn't think of. Like, we didn't think of this when we started these kinds of experiments. The other point I want to make is there can be density-dependent migration, like emergent density-dependent migration among animals, for sure, right? But there's no reason to believe that it has this hump-like shape like here, and that they would at some point jam up. No, probably not, right? So that's why this way it doesn't work, but it works as I think the questions that were asked in this field were so amazing. Like sometimes it really helps to go back in time, even before you were born, and read papers and just learn from those questions.
Cool, thank you.
Thank you for a nice talk. I have a question and a proposal for you. Have you done this with a different type of bacteria? Are these results repeatable with different types of bacteria?
Yes, Vibrio cholera, for example. Also for not all E. coli, for some E. coli, we saw that. There are also strange new phenomena that can occur, depending on the bacterium. I can show you one if you're interested. And also, we have done it only with ones that don't swim. Yeah.
Right. So my proposal is there is a very well-established symbiosis system between the squid and Vibrio fishery.
Yeah.
And the squid has crypts that look similar to these columns that you're showing here, and they have different sizes. A given crypt has different size, and usually they are colonized by different strains of Vibrio fishery. Now we know that if you grow the Vibrio fishery on a agar plate, different strains, even competing strains, they do coexist. But when they colonize the crypts of the squid, they do not coexist. One strain takes over. So if you were to introduce now these different strains in equal proportions from the beginning in a system like this, would one strain take over once they are in those artificial crypts that you're building here? And it's a model system. It's nice because we know that there is only one colonizer, one symbiotic bacteria that likes to sit in those crypts and not, and they do take over. So.
Well, thank you for the suggestion. I mean, this sounds definitely like something I would be interested in doing. I think, isn't that also a model system for quorum sensing, Vibrio fischeri? Right, so this is something we...
But before they do the bioluminescence, I mean, in the squid, they do bioluminescence in the squid, right?
Yeah.
And first, the problem is the recruitment.
Right, and during the recruitment, do they do quorum sensing already?
Not as far as I know.
Okay, okay.
They are present in very small cell counts,
Mm-hmm.
and it's enough to recruit a few cells to get to the entrance of the crypt and take over. Then we have the concentration of the cells is very small.
Okay.
And they still do take over.
Okay, yeah, would be very interesting to do. We are also really interested in quorum sensing in the system because that could really introduce something like an Ely effect, as was mentioned before. Yeah.
Okay, I have a question too, Oskar. So you mentioned a few times that you're using non-motile bacteria. Why is that?
I thought it would be really interesting to figure out if you bring in activity through this proliferating part, like locally injecting, degrees of freedom, etc., to keep the rest passive. Like on a fundamental level, I think that's interesting. What kind of pattern formation do you observe if, at least you can describe the movement purely passively, right? And this way we could go back to colloidal physics to ask what is this collective diffusivity as a function of density. If that is also active, then it gets an even harder problem. But I agree we should also do this with motile strains, and things will change because many things can change, but certainly the length scales have to be larger because the effective diffusivity would be larger. And they probably also interact with the surfaces in surprising ways. And so it's going to be much more complex, perhaps also much more interesting. Yeah.
And my second question is about the widths of those niche here. Like, how did you choose 50 micrometers? Why is that?
Yeah, well, initially this was just serendipitous, and then we varied it. Let's say a factor of 2 doesn't change. What we haven't done yet is systematically vary the width. If this becomes too wide, based on simulations, we expect it will, the bacteria will essentially see just one curving surface and will not be able to colonize at all. But there will also be some kind of transition happening probably. So we think it's also fruitful to vary that systematically. We just haven't done it yet.
All right, let's thank Oskar one more time.